Hello everyone, and welcome to General Microbiology, a channel that provides high-level introductory videos on everything micro in biology, from bacteria to viruses and other microscopic life. Today we will talk about viroids, virusoids, and satellites, or generally the subviral elements of life. As far as we know today, viroids, virusoids, satellites are the smallest elements of life, and are generally termed subviral life. These simple forms of subviral life may have very well been the origin of life itself prior to the existence of proteins and multi-component life forms such as viruses or bacteria. Using broad categories, a viroid is simply a short strand of circular, single-stranded RNA with no protein capsid or protein coding in their genetic material. To date, all known viroids infect higher plants. A satellite is also solely comprised of genetic material, but in contrast to viroids, codes for proteins. Finally, a virusoid is generally defined as a viroid that has been encapsulated with a coating of protein by a helper virus. However, this is where the definitions begin to merge. The hepatitis D virus, which is generally considered an encapsulated viroid, actually codes for protein. In its encapsulated form, it is considered a virusoid, and virusoids themselves are considered to be a subgroup of satellites. The genome of satellites may be comprised of DNA or RNA and range from approximately 220 to about 1,500 base pairs, although some satellites have been identified with genomes nearly 20,000 bases in length. While the genome of known viroids is generally smaller, at approximately 250 to 500 bases. Neither satellites or viroids have capsids or protein shells, while virusoids are generally encapsulated. Finally, viroids generally infect higher plants, while satellites and virusoids have been identified in bacteria and mammals. The boundaries of current definitions are changing and not clearly separated. As we identify new viroids, virusoids, and satellites, science may yet change the definitions further. Overall, however, subviral particles are by themselves comprised of genetic material, typically without a protein, polysaccharide, or phospholipid coating, until encapsulation occurs with the assistance of a helper virus, as in the case for virusoids. As one can imagine, viroids, virusoids, and satellites are extremely small in terms of size. Our cells are about 10 to 20 micrometers in diameter, bacteria a few micrometers in diameter, viruses are about 10 times smaller at about 50 to 100 nanometers, but the subviral particles are the absolute record holders in size at only about 5 to 30 nanometers. Similarly, in terms of genetic material, they hold as little as approximately 220 base pairs, which is only about 5 to 10,000 atoms. For comparison, an adenovirus has a genome of approximately 26 to 48,000 bases, and the genome of Yersinia pestis, the bacteria that causes the Black Plague, is over 4.5 million base pairs long. Satellites can fall into any of the viral categories, and every type of satellite has been identified. Single-stranded RNA, such as the tobacco necrosis virus, double-stranded DNA, such as the Sputnik virus, single-stranded DNA, including alpha satellites, double-stranded RNAs, such as Saccharomyces cerevisiae M virus satellite, single-stranded satellite RNAs, which themselves fall into three groups of large satellite RNAs, small linear satellite RNAs, as well as circular satellite RNAs or virusoids. Hepatitis D, which is a circular RNA satellite virus, which falls into category 5, is in itself a virusoid, which is a subclass of satellites, and starts out as a negative sense RNA strand that binds tightly to itself, forming a rod-like structure. The genome of Hep D virusoid is only about 1.7 kilobases long. Its genetic material is a negative sense single-stranded RNA. but due to tight base pairing between two regions, forms a rod-like complex which has been shown to be recognized by various mammalian DNA-dependent RNA polymerases. The circular RNA has a clearly defined cleavage site, a poly-A signal, a UAG to UGG editing site, and a delta antigen open reading frame. The genome undergoes self-cleavage to form the messenger RNA with a very similar structure that includes a 3' poly-A tail and a 5' cap. The infection cycle of the Hep D virus is relatively straightforward, but does have some peculiarities. The Virusoid is capable of infecting both dividing and quiescent cells, but only in its Virusoid form, that is, when it's encapsulated by the proteins from the helper virus, typically Hep B. The attachment to the NTCP bile transporter is mediated by a protein capsid, which also enables the next stage of receptor-mediated endocytosis. Following endocytosis, the genetic material is translocated to the nucleus and uncoded. The first surprising discovery was the ability of Hep D genetic material to utilize a DNA-dependent RNA polymerase despite only having a single-stranded RNA template. During this process, three forms of RNA are produced: circular genomic RNA, circular complementary antigenomic RNA, and the linear polyadentylated antigenomic RNA, which is in turn the messenger RNA and contains the open reading frame for HDAG, or the delta antigen. The delta antigen is in itself produced in two forms: the small 24 kilodalton form, which plays a role in viral replication, and the large 27 kilodalton form, which is required for assembly of the particles. The Hep D genome is only capable of producing HDAG, large and small proteins, as well as the ribozyme for genetic material processing. The genetic material of a helper virus is required to propagate viral replication, assembly, and export. When such a helper virus is present, a role most commonly played by Hep B, at times hepatitis C, a by itself non-pathogenic viroid becomes exceptionally virulent. A co-infection with Hep B and Hep D poses the highest risk of mortality among all hepatitis patients, increasing the risk of hepatocellular carcinoma threefold and mortality in general twofold. For contrast, infection of Hep B alone is typically cleared by the body in 70% of those infected, with more than 95% of the patients progressing to full recovery and development of protective immunity with a routine antiviral protocol, which is in itself only required for a very aggressive and rare form of fulminant hepatitis. Another interesting example of a satellite is a Sputnik virophage. This is a satellite virus that has been shown to inhibit the replication of its helper virus and thus act as a parasite of that virus. While not a true infection of its helper virus, the Sputnik satellite infects amoeba cells and hijacks the Viroid replication machinery to rapidly produce numerous copies of itself. The size of the Sputnik genome, which has been reported to be as large as 18,000 bases, with the density of the packaged DNA comparable to the T4 bacteriophage and the adenovirus, is also quite surprising. Finally, the recent analysis of the Sputnik genome serves supportive to the hypothesis of horizontal gene transfer between the mimivirus and the Sputnik, a phenomenon previously thought to occur only in cellular life. View of viroids, virusoids, and satellites, the three closely related and often intermixed categories of subviral life.